In Chapter 12, we are going to learn all about Gregor Mendel's experiments and the concept of heredity. So this is a background slide on Johann Gregor Mendel. He is considered the father of genetics, and that is basically all I really need you to take away from this slide. He's considered the father of genetics. All of this information here, this is just some background information on him about where he came from, what he did, what led him to doing the work that he did. We're going to talk about his work. We're not necessarily going to talk and have test questions on his background. So if you want to learn more about his background, you can read through this slide, and it's also mentioned in the course text. But for the purpose of this class, I want to focus on the work that Gregor Mendel did and the concepts of genetics and genetic inheritance. Gregor Mendel used the garden pea, or Pisum sativum, to study patterns of inheritance. And he used this species because it had many simple dichotomous traits. He could understand the flower color, the seed shape, and seed color amongst many other different traits. And many traits were the result of true breeding. And peas are known as monoecious species, which means there is a male component and a female component in a single flower, so it's very easy to manipulate matings and mate the exact flowers and the exact combinations that you would want. And what Mendel found was each pea was a new individual, so he could evaluate thousands of offspring and collect a lot of data. Breeding peas had a short generation time, and they were very cheap to do. It was very cheap to breed peas. They're relatively inexpensive and readily available. So excellent model system to study inheritance and inheritance patterns. Gregor Mendel performed what's known as hybridizations, which involve mating two true breeding individuals that have different traits. And this is the notation system that he used. So this slide is really important for you all to understand, because as we go through the breeding sequences and as we talk about Punnett squares and things, you are going to have to know this shorthand notation. So P indicates the parent generation of the true breeding plants. F1 is the first generation of offspring from a cross of parents. So this is going to be the brothers and sisters, the first progeny. F2 is the second filial generation of offspring produced by crossing two of the F1 individuals with each other. So P is the parents, this is going to be the first generation, this is going to be the second generation, and then all the Fs thereon afterwards are going to be the succeeding generations. So F3 is generation 4, F3 is generation 3, F4 is generation 4, and F5 is generation 5, and so on. Siblings, I'm sure many of you have siblings. You know what siblings are, brothers and sisters. Mendel did cross-sib mating, so he mated brothers and sisters of the same progeny to look at different traits and patterns of inheritance. And then he also did what's known as back-crossing, where he mated an offspring back to a parent of another ancestor. And he also did self-crosses, because the pea plant actually has a male and female component in the same flower. Mendel was able to do self-cross, which basically bred the plant with itself, also known as selfing. And true breeding, I mentioned this earlier, when parents have the same traits, always produce offspring having the same traits. And Mendel found that true breeding was a phenomenon that he observed in garden peas, which was another major benefit to him using this as a model system. So before we dive into all of the experiments that Mendel did, it's important for you all to understand some terminology so that when I use these words, you know exactly what I'm talking about. I'm going to be talking a lot about traits. So these are characteristics like flower color. So I can say the purple trait or the white color trait. Those are traits, those are characteristics, flower color, eye color, etc. Phenotype versus genotype. A lot of people confuse these two. I always remember phenotype has the P in it, so this is the physical appearance. This is the version of a trait that we actually see. So the phenotype of the flower is that it's a purple flower, or the phenotype of the flower is that it's a white flower, or the phenotype of eye color is that the person has blue eyes. Versus genotype, this is what we cannot see. These are the genes. This is the genetic composition of the person. So this is the specific combination of hereditary particles carried by an individual that cause a phenotype. So genotypes lead to phenotypes. Phenotypes are the physical expression. Genotypes are what the genetic sequence is actually made up of and causes. Alleles. So we're going to talk about dominant versus recessive alleles. Alleles are specific versions of a hereditary particle. So these are versions of different genes. So we're going to say they have dominant alleles or recessive alleles. So in the example I'm going to give you, when Mendel crossed the purple flower with the white flower, in the F1 generation, the first progeny, you're going to notice that purple is the dominant allele. We were going to see all purple flowers, and I'm going to explain why. So dominant, those are alleles that mask others, and they're often designated with capital letters. So the P for the purple flower, capital P. Recessive alleles, they are going to be the ones masked by others. They are recessive. They are not going to be the ones expressed in the dominant fashion. And they're often designated with lowercase letters. So we can use lowercase p for the white flower, the example I'm going to give you. So we've already done genotype. Now we're going to talk about homozygotes and heterozygotes. So homozygotes occur when both alleles for the same trait are the same. So if we have big P, we have a true dominant purple flower. When we have little p, we have a true recessive white flower. Homozygotes are always true breeding. That's a very important concept. Homozygotes are always true breeding. Heterozygotes, that's when alleles for the same trait are going to differ. So these are going to be your hybrids. So big P, little p. What's that going to give us? Is that going to give us a purple flower or a white flower? Or maybe something in between. We're going to talk about that. But heterozygotes, really important, are always non-true breeding. And you're going to see why in the example I'm going to give you with the purple and the white flower. And then the zygote, we've already been over what a zygote is in meiosis. This is the first diploid cell produced by fertilization. Some more useful terms that you need to know before we get into this lecture: what a gene is. So that's the place in the DNA strand that encodes information that causes a trait. The locus is the place of interest on a chromosome, usually a gene. Alleles, again, they're versions of the gene. So we have dominant alleles and we have recessive alleles. We're going to do a couple examples of what's known as reciprocal crosses. This is a formal mating cross where a previous cross is repeated, but the parents exhibiting versions of a phenotypic trait are reversed by sex. I'm going to give you direct examples of reciprocal crosses throughout this lecture. The wild type allele versus the mutant allele. Okay, the wild type is going to be the most common allele, the wild type. That's the one that's predominant in a population, so that's known as the normal allele. The mutant allele is the rare one. Mutants are rare. So these are the non-normal alleles. Non-normal alleles. So this slide depicts an example of a formal cross for one trait. So we're looking at true breeding parents here. So I'm going to name this one parent 1, and I'm going to name this one parent 2. So we're looking for if these are true breeders, are they going to yield purple flowers in all of their offspring? So again, this is the parent generation indicated by the capital P. F1 is going to be the first generation of offspring. And when we bred these two parents together, what we see are two purple plants. So these are true breeders in terms of color. And then what happened here was the siblings crossed, or there was sibmating. So siblings that inherited the traits and the genes from that parent generation, they bred in this cross F1, and they produced an F2 progeny here. And same thing, they got the same phenotype. We got purple flowers in each one. So these are true breeders for the purple flower phenotype. On this slide, we're going to talk about formal monohybrid crosses. So Mendel crossed parent plants that differed in one trait to produce what is known as hybrid strains in order to see if what was inherited could actually be detected. So he crossed true breeding purple with a true breeding white in the parent generation. So one expressing the purple phenotype, one expressing the white phenotype. And interestingly, in the F1 generation, they were all purple. There was no blending. There was no visible mix between the purple and the white plants. However, when he did sib mating in the F1 generation and he bred the two siblings that were the progeny from that parent generation, well, then he noticed he got 1, 2, 3 purple and 1 white plant. So he had this strange 3-to-1 ratio in the F2 generation. So the F2 sibs were both purple and white. So this was obviously not an example of true breeding, and this sparked Mendel's curiosity to further investigate this phenomenon. Now I want to break down that previous slide in a little more detail and add some of the terminology that you should all be familiar with now. So in the parent generation, we bred a purple homozygote, so big P, with a white homozygote, little p. Okay, and what did that give us in the first offspring generation, or the first progeny, that F1 generation? Again, all of the flowers turned out purple. There was no blending. But why did we get 100% purple? Well, because we got these heterozygotes, big P, little p, big P, little p. These heterozygotes, we know that the big P must be the dominant allele. So the big P masked the expression of the little p. So again, in the F1 generation, there was 100% of the progeny that was purple. And then when we did the sib mating and we bred these heterozygotes together, well, then we got a mix. So this is where our fun Punnett square comes in. So I don't know if you all have taken genetics yet or know what Punnett squares are, so excuse my drawing. So I put big P, little p. I put one genotype on top and I put one genotype on the side. And then I do a cross. So here I got big P, big P. Here I got big P, little p. Here I get big P, little p. And here I get little p, little p. So just as represented here. So I have one true breeding purple homozygote, and then I have non-true breeders, 2 heterozygotes, but they still express the purple phenotype. So these 2 here, still expressing the purple phenotype. And then I get 1 white homozygote. So the F2 sibs, that's why they produce both white and purple. And that combination of breeding the homozygotes and the heterozygotes and all those examples I gave you on the previous slide, the reason the offspring come out the way they do is due to Mendel's first law. So he says hereditary is particulate. Each true breeding parent carries 2 hereditary particles for each trait. So in that case, the hereditary particles for the homozygote purple were big P. The hereditary particles for the heterozygote purple were big P, little p. And the hereditary particles for the white were little p. Particles come apart during gamete formation such that each parent passes only one particle or the other of a pair into each of their gametes. And we talked about this in meiosis, how meiosis actually works. So that's how we get the 4 at the end of meiosis. Hereditary particles are then recombined randomly during fertilization to produce offspring. Again, on this slide, just some more examples of Punnett squares for you to practice with. So we're looking at the parental gametes. So we put one parent's gametes on top, and then we put the other parent's gametes on the side. So this parent on top, big P, what does this have to be? Well, that has to be the dominant purple flower. Little p, what does that have to be? Well, that has to be the recessive white homozygote. So we breed those together, we bring this p straight down, and then this p over. So we get a hybrid expressing purple. We bring this p straight down, we bring this p over. We get another hybrid, purple. We bring this p straight down, we bring this little p over. Another hybrid, purple. And then, same thing, we bring this big P straight down and this little p right on over. So they're all purple, aren't they? That's what we did in that F1 progeny example. And then again, when we cross the F2 siblings, so we're just taking, let's take this one, and let's write their alleles across the, or let's write their gametes across the Punnett square. So big P, little p, it's breeding with big P, little p. So then what do we get? Well, here we get big P, big P, big P, little p, big P, little p. And I always put the capital letter first, regardless of the order that it's in, just for clarity's sake. And then we get little p, little p. So what do we get? We get that 3-to-1 ratio. So these three are still going to express purple, and then this one is going to have the white phenotype. This is going to be a white homozygote. So we're going to have 2 purple heterozygotes, and we're going to have 1 purple homozygote and 1 white homozygote. Throughout Mendel's career, he did a lot of different test cross because he wanted to determine whether an organism expressing a dominant trait was a homozygote or a heterozygote. So he had all these different yellow P's, and yellow tended to be the dominant trait. Green was the recessive trait. But over the course of time, so many different breedings, he may have lost track. So he would do some test crosses to determine if the gametes, what the gametes were. So what he did here was he crossed an unknown Y with a known green. And what did he get? Well, he got 100% big Y, little y. So this test cross resulted in the finding that the original parent, this one right here, had to have been big Y, big Y. It's kind of like reverse genetic engineering. If we didn't know what the parents' alleles were, we could breed that with the completely recessive homozygote and we could find it out. And then look down here in this example, the second example. So gametes, same concept. So he crossed a yellow P with a green P. And well, this time what he noticed is that he got 50% yellow and he got 50% green. So we know that this one up here had to be a little y. Had to be a little y because this y had to have been brought down into that Punnett square, and then also had to have been brought down into that one. So this test cross resulted in this 1-to-1 ratio of yellow to green offspring, indicating that this original parent had to have been a heterozygote, had to have big Y, little y. So he figured out that each parent carries 2 alleles for each trait. Alleles segregate equally and randomly into gametes, and gametes randomly recombine alleles in zygotes during fertilization. And Punnett squares are really useful for predictions. They can predict Mendelian cross outcomes. So let's go through this monohybrid cross and we'll figure out what we can expect as far as the offspring. So again, monohybrid cross. We're crossing a dominant yellow with a recessive green. So 2 dominant alleles, 2 recessive alleles. And what we get here in the offspring, so remember this parent can only give one of its alleles, or one gamete. So it gives the big Y. This parent can only give one of its gametes, or one of its alleles. So it gives the little y. And that F1 generation, then we get that heterozygote yellow. So it's still going to be yellow because the dominant allele is going to mask the recessive allele. And then what does this parent do when it gives off its alleles or gametes? It's going to give one big Y and it has to give one little y. And let's say we did a SIB cross of this F1. Let's say there's another big Y, little y. And even though that P is going to be expressing yellow, when you do the SIB cross, wow, we're back to that 3 to 1. It's a terrible 3. Let me erase that. Sorry about that. We're back to that 3 to 1 ratio that we saw in the flower crosses when we bred the purple flower with the white flower. And so we can get down here into our ratios. So phenotypes, again, that's the physical traits, so that's what we can see. So the yellow peas have a yellow phenotype, the green peas have a green phenotype. The genotypes are their alleles, what they're made of. So big Y, that's going to be the dominant yellow pea, or the homozygote. Big Y, little y, that's going to be the heterozygote. Still a dominant phenotype is going to be expressed. Little y, that is a homozygote recessive. And then we look at the genotype ratio. So let's look at the genotype ratio that was expressed in the F2 progeny. So we have YY, big Y, only one of them. We have big Y, little y, we have 2. And then we have little y, 1. So we have this 1 to 2 to 1 ratio. That's going to be a very important ratio, and you're going to see that ratio repeat time and time again, the 1 to 2 to 1 and the 3 to 1 ratios. So before we move any further, I want to talk about some general probability basics. I'm sure many of you are already familiar with probability and what probability means, but chance events are described by probability. So probability is given as a fraction or a decimal equivalent over a range of 0 to 1. Probability of 0 means an event will not happen, and therefore it's not due to chance at all. A probability of 1 means an event will happen. It is certain and therefore not due to chance at all. Anything that is chance ranges between those values. So let's think about flipping a coin. Well, what are the chances that you get heads? What are the chances that you get tails? So you flip a coin 100 times, 1 out of 2, or 50 out of those 100 times, theoretically, you should get heads. 50 out of those 100 times, theoretically, you should get tails. So that would be 0.5. Convert 0.5 into a percent, you get 50%. Probability basics. I'm sure you're all familiar with this already. Now let's talk about the multiplicative law of simple probability. So many of you who have grown up playing the board game Monopoly may be familiar with this law of simple probability. So what do you have to do in Monopoly to get out of jail? Well, you can either wait a certain amount of turns or pay a certain amount of money, or you can get lucky and roll 2 6s. So what are the chances of you rolling 2 6s on that first attempt? So that has to do with the multiplicative law of simple probability. So the overall chance for the occurrence of 2 or more independent random events in exact order is equal to the product of their individual probabilities. So what does that mean? So let's say we have a 6-sided die. No matter what the number is on that 6-sided die, you have a 1 in 6 chance of rolling that number. So you have a 1 in 6 chance of rolling a 5, a 1 in 6 chance of rolling a 4, a 1 in 6 chance of rolling a 6, so on and so forth. So let's say here, I'm going to change this to 6 because we want to get out of jail in Monopoly. The overall chance of rolling exactly a 6 on the first die and then a 6 on the second die is what? How do we figure that out? Well, first die we have a 1 in 6 chance. Second die we have a 1 in 6 chance. That's going to give us a 1 in 36 probability. So not a very good probability. But same thing here. You know, if I wanted to roll a 5 and I wanted to roll a 4, just as the vignette originally stated, it would have the same probability, 1 in 36. So it might be better to skip the turns or pay the fee, but if you're lucky and you roll that 1 out of 36, there you go, you're out of jail. So we talked about the multiplicative law of simple probability. Now let's go into the additive law of simple probability. So when there are multiple ways or tries for a chance outcome to occur, the overall probability is the sum of the individual events. So let's give this example a try. You want to win a game that you're playing with your friend. You can win this game by rolling either a 4 or a 5 with 1 die. And then what's your overall chance of winning? So on a 6-sided die, if you get 2 of those 6, well, then you have 1/3 chance, right? So the chance of rolling a 4 is 1 in 6, or the chance of rolling a 5 is also 1 in 6. So the overall chance of rolling either a 4 or a 5 is 1/6 plus 1/6, which is going to give us 2/6 or 1/3. So both the laws we just talked about, the multiplicative law and the additive law, can be combined for complex random events. So let's give this example a try here. And again, we're still using that 6-sided die. So let's talk about rolling a 5 and a 4 in any order. And what are the chances of that? So on a 6-sided die, the chance of rolling a 5 first is 1 in 6. Then we times the chance of rolling a 4 on the second die, 1 in 6. So we've already done that example, 1 out of 36. The odds aren't in our favor. But if order doesn't matter and we want to see if we can roll a 5 and a 4 first or a 4 and a 5 first, then we have to combine the rules that we've learned. So we could roll a 4 first. That's another 1 in 6 chance. And we could roll a 5 second, another 1 in 6 chance, leaving us with another 1 in 36 chance. So again, we use the multiplicative laws here, and now down here we're going to use the additive law. So the overall chance of getting a 5 and 4 is in any order. All we have to do is add what we found from the multiplicative law together. So 1 out of 36 plus 1 out of 36 gives us 2 out of 36, and then we reduce it to simplest terms, and we get 1 out of 18. So better than that 1 out of 36, but still the odds are more likely against you that you're not going to be able to do that. Now I want to talk about pedigrees. So before we get into talking about the pathologies and the diseases, it's important for you to understand the symbols that are used for representation purposes in pedigrees. So males are represented by squares and females are represented by circles. And unknown genders are represented by diamonds. So this is going to be our P, this is going to be our F1, and this is going to be our F2 down here. So whenever the shapes are filled in, like we see here, that means that person is affected by whatever disease that is affecting them. So this male, and again you can see male symbols depicted like this, and you can see female symbols depicted like that. So an affected male breeds with an unaffected female. So this one unaffected minus, and this is an affected male. And they have babies. What are their offspring? Well, we got one male who's unaffected, we got one female who's affected, and we've got one unknown gender who is unaffected. And then this male is going to breed with a different female. Both are unaffected. What are their progeny going to be? Well, again, 50-50 chance of having males or females. And they had 2 healthy females, so 2 unaffected females. If there is a line stricken through this, so let's say right after birth, unfortunately, this mother died. well then if there's a line stricken through it, that would represent that that female was deceased. Now we're going to use a pedigree to better understand our first pathology that we're going to talk about, known as Albinuria. This is also known as black urine disease. This is a very rare inherited disorder that prevents the body from fully breaking down amino acids, the amino acids specifically tyrosine and phenylalanine. And it results in a buildup of a chemical called homogentic acid in the body. So with albinuria, let's begin by seeing who's affected and who's not affected. So in generation 1, we have an affected male and we have an unaffected female. In the second generation, we are going to have an affected male and a heterozygote unaffected female. So again, this is a recessive disease. So it's only expressed when the recessive alleles are able to fully express themselves. So that's why you see little a being affected, and you see the heterozygote here not expressing or not affected. So this one up here, number 1, had to have been little a, and this one here had to have been big A, little a, because there has to be a way for this male to get both recessive alleles. So we figured out 1 and 2. Now, the affected male breeds with the unaffected female in the second generation. And what do we get? Well, then we get this third generation right here, 1, 2, and 3. So we have little a breeding with big A, little a, and that's going to give us big A, little a, big A, little a, and then all little a's, little a, little a, little a, little a. So this one, what does this have to be? What does this unaffected male have to be? Well, he has to be one of these two. It's the only possible combination that he could possibly be. So he has to be a heterozygote. So I'm going to fill this in. Big A, little a. And then we have an affected female over here. So this affected female, when she breeds with this unaffected male out here, she has an affected child. So the only way for her to have an affected child is this male has to be an unaffected heterozygote. Now let's figure out this one down here. So I can give you an example of a pedigree like this on your quiz or your test, and I can pick any one of these circles or squares and have you identify the alleles. And you may also have to tell me whether they're unaffected or affected. So over here, we're looking at big A, little a, breeding with big A, little a. And in this fourth generation, what do we get? So again, let's do that. Punnett square. Big A, little a. Big A, little a. That's going to give us big A, big A. Big A, little a. Big A, little a. And little a, little a. So we have an affected female here. And from this information, unless we had more of this pedigree, we really can't decipher what this is. This could be big A, because there's a 25% chance of that. Or this could be big A, little a, because there's a 50% chance of that. And in both cases, that female would be unaffected. So we need more information to determine that one. Now we're going to move into talking about a formal dihybrid cross. So what we're going to be looking at here is multiple traits. So the P generation, or the parent generation, what we're going to do is we're going to cross parents that were true breeding for different phenotypes of two different traits. So to be true breeding, what does that mean? Well, they have to be homozygotes. So they have to be homozygotes for each of the two different traits. So here what we're going to look at is one of Mendel's famous experiments where he figured out this ratio, this 9 to 3 to 3 to 1 ratio that we're going to talk about. And he was looking at the seed shape, whether the seeds were round or wrinkled, and he was looking at the seed color, whether the seeds were yellow or green. All right, so the P generation, the parent generation. Again, we have to have homozygotes for both of the traits. So big Y, little y, that's going to give us the yellow color, and then big R is going to give us the round color. Little y is going to give us the green color, and little r is going to give us the wrinkled phenotype. When these two breed in the F1 generation, what do we get? They are heterozygotes. These are dihybrids. So we're going to get big Y, little y. We're still going to be expressing that yellow color. The yellow is dominant. And we are still going to get a round seed. You can see the nice round shape here. This one's wrinkled. So we get the round seed because we have big R, little r. And the big R is masking the phenotype of the little r. So then let's look here. Let's look at the gametes from heterozygous parents. And we're making all the possible combinations of gametes that we possibly can. So we're taking big Y, big R. We're taking little y, big R, big Y, little r, and little y, little r. And we're breeding that with the same combination of heterozygote gametes here. Big Y, big R, little y, big R, big Y, little r, little y, little r. And I'm not going to go through all of this, but what we do is we get a 9:3 to 3 to 1 ratio of phenotypes in the F2 generation. So we get 1, 2, 3, 4, 5, 6, 7, 8, 9 offspring that are yellow and round. We get 1, 2, 3 offspring that are green and round. We get 1, 2, 3 offspring that are yellow and wrinkled. And we get 1 offspring that is green and wrinkled. So that's the 9 to 3 to 3 to 1 phenotype. So I encourage you all to write out this Punnett square, make sure that you can bring these alleles down where they need to go, and you get this proper ratio of 9 to 3 to 3 to 1 in the F2 of the dihybrid cross. You get this proper Mendelian ratio. So from the previous experiment, the previous slide that we just talked about, if you count for each trait individually, you will see each still gives that 3 to 1 ratio that we saw in the monohybrid crosses. So this led Mendel to conclude that traits were inherited independently from one another. Traits were inherited independently from one another. And this led him to formulate his second law, the law of independent segregation. So segregation and recombination of alleles for one trait has no effect on segregation and recombination of alleles for other traits. Okay, so each trait is exclusive and has no effect on the other traits. So that's one of the explanations he put forth to explain why we got the 9 round yellow, the 3 round green, the 3 wrinkled yellow, and the 1 wrinkled green seed. Mendel's results that traits are inherited as either dominant or recessive pairs contradicted the view at the time that offspring exhibited a blend of their parents' traits. However, the heterozygote phenotype occasionally does appear to be intermediate between the two parents, and that's exactly what we see in snapdragons. So cross a true breeding red flower parent with a true breeding white flower parent. And what do we get in snapdragons? All pink F1 heterozygotes. So all pink F1 heterozygotes. And then if you cross the pink F1 sibs, you get an F2 that has the following ratio of phenotypes. Then we're back to that 1 to 2 to 1 ratio. So we get 1 red, 2 pink, and 1 white. This is an example of incomplete dominance. So this incomplete dominance denotes the expression of 2 contrasting alleles such that the individual displays an intermediate phenotype. The allele for red flowers is incompletely dominant over the allele for white flowers. However, the results of a heterozygote self-cross can still be predicted, just as with Mendelian dominant and recessive crosses. Mendel implied that only 2 alleles, one dominant and one recessive, could exist for a given gene. But we know now that this is an oversimplification. Although individual humans, and all diploid organisms for that matter, can only have 2 alleles for a given gene, multiple alleles may exist at the population level such that many combinations of 2 alleles are observed. So when many alleles exist for the same gene, the convention is to note the most common phenotype or genotype among wild animals as the wild type. And this is considered the standard or the norm. All other phenotypes or genotypes are considered variants of this standard, meaning that they deviate from the wild type. The variant may be recessive or dominant to the wild type. And that's exactly what you see in this picture here. There's multiple alleles that deal with the coat color in the rabbits. So 4 alleles exist for the C gene. The wild type version, C++, is expressed as brown fur. The chinchilla phenotype, or little c, ch, little c, ch, is expressed as black tip white fur. That's the chinchilla right here. The Himalayan phenotype, little ch, has black fur on the extremities and white fur elsewhere. And then finally we get into the albino rabbit. The albino rabbit has the colorless phenotype, and that's denoted by little c, and you see the white fur. So in cases of multiple alleles, dominance hierarchies can exist. In this case, the wild type allele is dominant over all the others. Chinchilla is incompletely dominant over Himalayan and albino, and Himalayan is dominant over albino. This hierarchy or allelic series was revealed by observing the phenotypes of each possible heterozygote offspring. In certain cases, one mutant allele can be dominant over all other phenotypes, including the wild type. This may occur when the mutant allele somehow interferes with the genetic message so that even a heterozygote with one wild type allele copy expresses the mutant phenotype. One way in which the mutant allele can interfere is by enhancing the function of the wild type gene product or by changing its distribution in the body. And one example of this is the antennapedia mutation in the fruit fly Drosophila melanogaster that you see in the picture here. In this case, the mutant allele expands the distribution of the gene product, and as a result, the antennapedia heterozygote develops legs on its head where its antenna should be. Malaria is a parasitic disease in humans that's transmitted by infected female mosquitoes. And it's characterized by high cyclic fevers, chills, flu-like symptoms, and severe anemia. The 2 most common causes of malaria are Plasmodium falciparum and Plasmodium vivax. And Plasmodium falciparum is the most deadly, and that's what we see in the picture here, in picture B. This is Plasmodium falciparum, and this is, in figure A, this is Anopheles gambiae, or the African female mosquito that acts as the vector to transmit the parasite that we see in B into humans. When promptly and correctly treated, falciparum malaria has a mortality rate of about 0.1%. However, in some parts of the world, the parasite has evolved resistance to commonly used malaria treatments, so the most effective malarial treatments can vary by geographic region. In Southeast Asia, Africa, and South America, Plasmodium falciparum has developed resistance to the antimalarial drugs chloroquine, mefloquine, and sulfadoxone pyrimethamine. Plasmodium falciparum, which is haploid during the life stage in which it is infectious to human, has evolved multiple drug-resistant mutant alleles of a particular gene known as the DHPS gene. And varying degrees of particular drug resistance are associated with each of these alleles. Being haploid, Plasmodium falciparum needs only one drug-resistant allele to express this trait. In humans, as well as in many other animals and even in some plants, the sex of the individual is determined by sex chromosomes. The sex chromosomes are one pair of non-homologous chromosomes, and until now, we have only considered inheritance patterns among non-sex chromosomes, or autosomes. But in addition to 22 homologous pairs of autosomes, human females have a homologous pair of X chromosomes, whereas human males have an XY chromosome pair. So when a gene being examined is present on the X chromosome but not on the Y chromosome, it is said to be X-linked or sex-linked. And we're going to talk about the eye color in the model organism Drosophila melanogaster, or the fruit fly. So eye color in Drosophila was actually one of the first X-linked traits to be identified back in 1910 by Thomas Hunt Morgan. Drosophila males, just like humans, have an XY chromosome pair, and Drosophila females are XX. So in flies, the wild type color is denoted by big X, superscript big W. And it's dominant to the white eye color, which is denoted by big X, superscript little w. Because of the location of the eye color gene, reciprocal crosses do not produce the same offspring ratios. Males are said to be hemizygous because they have only one allele for any X-linked characteristic. Hemizygosity makes the descriptions of dominance and recessiveness irrelevant for XY males. Drosophila males lack a second allele copy on the Y chromosome, so their genotype can only be one of the two. They can have X, big W, Y, or they could be X, little w, Y. That's all males could be. In contrast, females have two allele copies of this gene. So females could be X, big W, X, big W. They could be X, big W, oops, X, little w. Or they could be X, little w, X, little w. So let's make that example relevant to humans, and let's look at how inheritance of sex-linked traits occur in humans. So the son of a woman who is a carrier of a recessive X-linked disorder will have a 50% chance of being affected. A daughter will not be affected, but she will have a 50% chance of being a carrier like her mother. So let's look at this pedigree here. So this is an unaffected father here. He's XY. And this is an unaffected yet a carrier mother. And that's denoted by the split of colors. So she's represented by half blue, half orange. So the males are either going to be unaffected like we see here, or affected from this generation. The females have no potential to be affected, but the females can be carriers and they can pass it on to their offspring. So we get one unaffected carrier daughter, and we get one affected completely healthy or normal daughter. And some alleles are so non-functional that they can actually kill the individual inheriting it. Lethal alleles can be dominant or recessive. And what we see here is known as a Manx cat. You see one blue eye, one off-colored brown hazel eye. All Manx cats are heterozygotes for the ML, or the M lethal allele. MM homozygotes, those are normal house cats. MML heterozygotes are all Manx cats that you see in this picture here. And if you inherit 2 copies of the lethal allele, they all will die. So there's no such thing as a viable M capital L cat. Crossing 2 Manx cats produce an offspring ratio of 2 Manx and 1 normal. Why does this happen? This is what I want you to think about. How many offspring were there? Did any of them die? Is this the proper ratio? Let me know. Chromosomal crossover can also be responsible for segregating genes. The process of crossover or recombination, we've talked about this in meiosis already, occurs when 2 homologous chromosomes align during meiosis and exchange a segment of genetic material. And then we get these recombinant chromosomes. So this chromosome and this chromosome, again, are going to be considered the recombinant chromosomes. And this chromosome and this chromosome are going to be considered the non-recombinant chromosomes. So in this picture, the alleles for gene C were exchanged. Gene C is at the bottom, and we see those alleles getting exchanged. The result is 2 recombinant and 2 non-recombinant chromosomes. In epistasis, the interaction between genes is antagonistic, such that one gene masks or interferes with the expression of another. Epistasis literally means standing upon. The alleles that are being masked or silenced are said to be hypostatic to the epistatic alleles that are doing the masking. Often the biochemical basis of epistasis is a gene pathway in which the expression of one gene is dependent on the function of a gene that precedes or follows it in its pathway. And the example of epistasis that we're going to talk about on this slide is about pigmentation in mice. So the wild type coat color, agouti, is depicted by big A, big A. This is dominant to solid colored fur, which is depicted by little a, little a. However, a separate gene at a separate locus, denoted by the capital letter C, is necessary for pigment production. A mouse with a recessive C allele at this locus is unable to produce pigment and is albino regardless of the allele present at the locus. Therefore, these genotypes, I'm going to write these out. So, oops, big A, little c, big A, little c, and little a, little c. All of those genotypes are going to produce albino mice regardless of the combination. A cross between heterozygotes for both genes. So let's do the cross. Do I have it depicted? No, I do not. Okay, so let's do the cross for the heterozygotes. So big A, big C, little c, big A, big C, little c. I want you to cross those and find out what you would get. This would actually generate an offspring with a phenotypic ratio of 9 agouti, 3 solid, and 4 albino. So in this case, what gene is epistatic? What gene is hypostatic? Well, hopefully you told me that the C gene is epistatic to the hypostatic A gene. So again, such multiple gene interactions produce modified Mendelian dihybrid cross F2 ratios. And in this case, this F2 ratio is going to be 9 to 4 to 3. So I want you to work through this Punnett square, try these different combinations, let me know what you come up with, cross these genotypes, and let me know if you can come up with this proper phenotype. 9 out of 16 should be agouti, 3 out of 16 should be black, and 4 out of 16 should be albino.